And now to our second lecture on amines. Amines can also do conjugate addition, which we introduced back at the end of chapter 18. Thus, if I have some kind of carbonyl compound that has an alkene adjacent to it, this type of compound is called an alpha-beta unsaturated aldehyde or ketone. And I introduce an amine as a nucleophile. The amine can come in at the beta carbon, thrust these electrons up, and then protonate this alpha carbon to give you this beta added aminoaldehyde. I now want to begin exposing you, and I mean that in a non-creepy way, to heterocyclic compounds, beginning with this slide. And what is a heterocycle? By definition, a heterocycle is a cyclic compound in which there is more than one kind of atom found inside the ring. For example, these three cyclic compounds are all heterocycles because each of them contains 4 carbon atoms and an additional atom other than carbon, either nitrogen, oxygen, or sulfur in the ring. There are lots of different nitrogen-containing heterocycles. One of these is pyridine, which you might remember from back in chapter 10. Because it meets all of the rules of aromaticity that we discussed in chapter 15, pyridine is aromatic. The nitrogen lone pair, which I haven't drawn here, is not released into the aromatic system because it is perpendicular to these pi electrons in the ring. These resonance structures here show us how the nitrogen in pyridine has a partial negative charge, which means that it actually withdraws or sucks electron density from the remaining carbon atoms in the ring. Now, because pyridine's nitrogen atom is electron withdrawing, electrophilic aromatic substitution reactions on pyridine occur at the meta position relative to the nitrogen. Thus, we could say that pyridine reacts similarly to nitrobenzene during an electrophilic aromatic substitution reaction. So here's our nitrogen in pyridine. I do an electrophilic substitution reaction. The electrophile will get added at the position meta relative to the nitrogen, similar to the way we would see in the electrophilic aromatic substitution of nitrobenzene. There are various other nitrogen heterocycles that I want you to have met. Not that I'm going to ask you any in-depth questions about them on an exam, but because I don't want them to look completely foreign to you when you come across them later in your careers. This compound, found at the core of numerous natural molecules, is called quinoline. Its structural sibling, shown here, is called isoquinoline. Now, isoquinoline should look familiar to those of you who are in my lab class because it is found at the core of this potent anti-leukemia molecule, AM636, which we discussed in lab as a potential group synthesis project. Note the isoquinoline core buried right here inside the molecule's lower left-hand ring structure. Another highly significant nitrogen heterocycle shown here is the porphyrin ring system. Why is it so important? Because this ring system constitutes the active site iron core of heme proteins, including hemoglobin, which our bodies use to transport oxygen to all of our tissues. Thus, we can see in the core of hemoglobin proteins, it has this heme active site in which these nitrogens present in the porphyrin ring system chelate an iron 2, which can be reduced or oxidized as it captures and releases oxygen from that we breathe. Because I don't want you to leave this class without hearing the word alkaloid, I am now going to say it with a funny accent. Alkaloid! Alkaloids are a special class of naturally occurring amines. A few famous examples of these are shown here, which you're welcome to remember if you so desire. This is an example, atropine, which is an antispasmodic alkaloid. This is a compound called ephedrine. And this is an antihypertensive drug known as reserpine. This brings us to one of the most important family groups of nitrogen heterocycles on the planet, the purines and pyrimidines. Now, purines and pyrimidines have the basic structure shown here. As you may remember from biology, adenine and guanine are purines, while cytosine, uracil, and thymine are pyrimidines. These 5 nitrogen heterocycles are the 5 nucleotide bases found in DNA and/or RNA. I have a special personal way of distinguishing purines from pyrimidines. Structurally, I like to remember that purines are structurally larger. You'll notice that they have more atoms than pyrimidines in their core. The way I remember that is remembering that the word purine is smaller than the word pyrimidine. Thus, purine, which is a smaller word, corresponds to the structure that is larger, and pyrimidine, which is a larger word, corresponds to the structure that is smaller. Now, I realize that that might seem absolutely insane and like an extremely stupid way of remembering this, but for whatever reason, when I was learning these structures back as an undergraduate, that's the way I remembered, and it stuck. Now, in order to remember the names of the purines and pyrimidines, I remember that adenine and guanine, the 2 purines, begin with the letters A and G, which spell ag. Because I'm a USU Aggie, and Aggies are pure in heart, it's easy to remember that adenine and guanine, AG, ag, are purines, pure in heart. Now the pyrimidines, cytosine, uracil, thymine, begin with the letters CUT, or cut, while an AG, adenine and guanine, or ag, or Aggie, is pure. An infected cut, cytosine, uracil, or thymine, is not. Thus, our cut heterocycles are pyrimidines. A great source of confusion that I faced as a young science student came from my inability to make the connection between how these nucleotide bases were actually related to the structure of DNA and how that was related to the structure of chromosomes. I'd learned from my many biology teachers that DNA contains the information that codes for all of our physical traits. I had also learned that through meiosis these traits are passed on with recombination to reproductive cells, and that through mitosis they are passed on to duplicating cells during eukaryotic cell division, all through the division of chromosomes. What I lacked, however, was the ability to see the connection between chromosomes and DNA. Do chromosomes contain DNA, I wondered? Is DNA located somewhere else in the cell, or is it in the chromosome, and if so, where? Now, to help reduce this confusion for you, I've assembled a few slides that you'll hopefully find illuminating. As you can see in this figure, purines and pyrimidines hydrogen bond. So right here, I have adenine and thymine, and down here I've got guanine and cytosine. Once again, these purine and pyrimidine pairs match up and they hydrogen bond in the complementary way shown here. Now in DNA, each purine and pyrimidine base is attached at carbon 1, you see the C1 prime here, to a 5-atomed ring sugar, as shown here. These sugars are all bound together in a strand by a phosphate backbone. So this little circle here is representing the phosphate backbone. Thus we could say that each DNA strand, here's a strand to the right and here's a strand to the left, consists of nucleotide bases that are linked together by a sugar phosphate backbone bound to C1 of the nucleotide base. The 2 DNA strands then have complementary base pairing. Adenine hydrogen bonding to thymine, and guanine hydrogen bonding to cytosine. Now if we zoom out in perspective, this sugar phosphate backbone looks kind of like this. A twisting pink ribbon with colored shapes representing the complementary nucleotide base pairs in between. This twisting ribbon structure is DNA. Now our cells package DNA by wrapping it around special proteins called histones, which act kind of like a wheel that we might use to wrap our garden hose around at home. These histones, in turn, are clustered together in groups called nucleosomes, which are then wrapped around in a larger structure shown here, which is called chromatin fiber. chromatin fiber, which is made up once again of numerous histone clusters wrapped up with DNA, then coils to form the chromosome superstructure shown here. So this is a summary of how packaged DNA really does make up the larger chromosome structure. As you may remember from biology, human somatic cells, non-reproductive cells, contain 46 chromosomes, while our reproductive cells, sperms and eggs, contain 23 chromosomes. One question you might ask is, how are our individual cells able to find a specific sequence of DNA in all of this mess, and then unravel it, and use it to make a specific protein right when they need it? That is an amazingly cool question. But I won't give you the answer until I teach you biochemistry during a later semester. Sorry. Now, the DNA Learning Center, the Cold Spring Harbor Laboratory in Laurel Hollow, New York, has produced a series of incredibly useful videos for teaching molecular genetics. I find one particular video of theirs to be most instructive for our current subject, how a chromosome structure actually relates to the individual sequence of nucleotide bases in our DNA. Now, I've posted the HTML here for you to examine later at your leisure. However, I'm going to show it to you right now just so that you can see it. Now, if we look at this video, you'll see these little white cylinders are complementary nucleotide bases, and they're being twisted around by this pink phosphate backbone. This giant purple blob here is a histone. These histones are being twisted around to form these larger histone groups around which the DNA has been wrapped. These then coil around to form chromatin, and the larger chromatin supercoils are twisted around and eventually wrap around and around and around until they constitute the chromosome superstructure. You'll see that being depicted here in the video, and I think it's supremely cool. And these chromosomes are what we actually see in a microscope when we're looking at a cell. As always, I conclude our video presentation with the promise that we'll see and have to do numerous synthesis examples together during our in-class problem sets. I look forward to seeing you then. And now I finish this lecture and this chapter by quoting from Dr. Seuss's classic book, One Fish, Two Fish, Red Fish, Blue Fish. My hat is old, my teeth are gold, I have a bird I like to hold. My shoe is off, my foot is cold. My shoe is off, my foot is cold. I have a bird I like to hold. My hat is old, my teeth are gold. And now my story is all told. Barbara Streisand.